the autonomic nervous system. Autonomic means automatic. But let me put it into a larger context. Let's look at the nervous system in general. And we have what is called the central nervous system and the peripheral nervous system. You, in laboratory, will spend time in detail talking about the anatomy of the central nervous system. These are the nerves that are protected in a bony edifice. So the brain and spinal cord are all encased and protected in the calvarium and in the spinal column. From the brain and from the spine, nerves go out. They also come back in, but where are they going? They are going to the periphery. They're going out to your fingertips. So from your spine, there's a nerve, that axon, that projects out to your fingertips, which helps in controlling muscles of your arms. There are nerves in your fingertips that go back to the spinal cord, taking you information, tactile information, temperature information, pain information. So this exchange then occurs between the central system and the peripheral system. The autonomic nervous system is comprised of both a central and a peripheral component. So let's look at those. We've got the sympathetic nervous system versus the parasympathetic nervous system. Anatomically, these two nervous systems are distinguished by where the nerves arise from the central nervous system. The sympathetic nervous system, cells arise from the spinal column in the thorax and lumbar region of the spine. The parasympathetic nervous system, cell bodies arise from the cervical region and sacral, which is down here. Each of these nervous systems is what we call a two-neuron nervous system. Now, there's obviously more than two neurons, but it is the anatomy and orientation of these nerves. So I'm going to draw a two-neuron system. Here's a cell body, an axonal projection, a synapse, and a second neuron. So there's a two-neuron system. Anatomists, microanatomists and neuroanatomists, if you have a large collection of cell bodies in the central nervous system, it's called a nucleus. So let's encase these cells in the spinal column. And this collection of cell bodies would be called a nucleus. A collection of cell bodies in the peripheral nervous system is called a ganglion. The cells can then take on another name. The first cell, the cell that arises out of the central nervous system, can be referred to as the preganglionic cell. And the cell arising after the ganglion is the postganglionic cell. So where do these ganglia occur in the two nervous systems? In the sympathetic nervous system, the ganglia lie very close to the spinal column. In fact, one of the concerns in spinal surgery is when you're in there whacking around bone that you have these collection of nerve cell bodies. And disruption of the cell of the nerves is much more difficult to treat than disruption to the bony components of the vertebra. So you always have to be very careful. These are anatomically very close. So for and there's one exception that we'll talk about later, the sympathetic nervous system has these ganglia very close. That means the preganglionic cell of the sympathetic nervous system is very short. The postganglionic cell, the axonal projection, very, very long, and it goes out to the organ of interest or the tissue of interest. These ganglia tend to be connected and share information between them. They occur very close, and they're enlargements because they're collections of cell bodies, so it's sometimes referred to as the sympathetic chain ganglion. There is this long chain of sympathetic ganglion that follow along the vertebral column. Contrast that with the parasympathetic system. The parasympathetic nervous system has long preganglionic cells. In fact, in most instances, the ganglia are located in the tissue of interest. So, for example, here is a preganglionic sympathetic that goes out to the heart, innervates the heart. So, different anatomy, different functions. Sympathetic nervous system activate flight or fright, emergency situations, conditions in which you want to get the body safely away. Parasympathetic, kind of the vegetative. Things are cool, things are relaxed, life is good. So let's look at some of these functions. When the sympathetic nervous system activates, it produces a dilation of the pupils. The pupils get large. Classic fear response. Kiddie entertainment, scare the cat, pupils dilate, very, very large. Parasympathetic, just the opposite, constricts the pupil. Very relaxed, small pupil. Salivary glands. You get that stage fright, the nervous anticipation. Maybe you've done something that's really hair-raising, right? Sympathetic nervous system inhibits salivary secretion. That kind of dry mouth. Parasympathetic, vegetative. You're going for the pizza. You want a lot of salivation. Heart rate, heart function. The sympathetic nerves produce an increase in rate and an increased force of contraction. You want to provide blood. You need it to escape. Parasympathetic, relax, decrease heart rate. GI tract. Don't need the GI tract in an emergency situation. Inhibit it. Sympathetic decreases secretions, decreases motility, shutting the system down. Parasympathetic increases it. Liver. Sympathetic nervous system acts on the liver to increase the amount of glycogen breakdown. You are providing glucose to the metabolizing tissues that need it. Parasympathetic stimulates glycogen production. You're post-absorptive, you just finished your pizza. Let's put that glycogen back on board. Intestines, colon. Don't need the intestines or the colon during a flight or fright situation. Sympathetic nervous system inhibits it. Parasympathetic stimulates it. Bronchi. Smooth muscle of the trachea and bronchus. Sympathetic dilates the bronchioles. That makes air movement easier. What do you want to do in a flight or fright situation? You want to have more substrates, you want to have more glucose, you want to have more air, so you dilate the pathways, and facilitates, it makes it easier to go in. Parasympathetic constricts it. What about blood vessels? Now here you see there's only a sympathetic arm that goes out to the blood vessels. And what it says, it constricts most blood vessels. There are important exceptions to that. And they should all make sense to you. Blood vessels to the kidneys, blood vessels to the gut. How important are those in a flight or fright situation? Not very. So, constrict. Sympathetic system, nervous system produces a vasoconstriction of these vessels. This vasoconstriction reduces blood flow. So you decrease blood flow to kidneys, to the gut. Normally, these can have very large, profound blood flows. 30% of your total blood flow at rest in the vegetative state goes to the kidneys. Don't need that in a flight or fright situation. So by shutting down blood flow to those tissues, you can redirect it to tissues that need the blood, that need the delivery of nutrients, that need the delivery of oxygen. Sympathetic nervous system produces a vasodilation of blood vessels in skeletal muscle, blood vessels in the heart. What two tissues are really going to need a lot of blood during flight or fright? Heart and skeletal muscle. So here are important exceptions. Adipose tissue, fats. Vegetative state, you want to store up fats. Exercise, you want to mobilize them. The sympathetic nervous cell stimulates the activity of an enzyme called lipase. Lipase breaks down lipids into fats. Fats which are mobilized and go to the muscle, skeletal and heart, to help support increased metabolism. Indeed, you'll see that metabolic rate goes up when sympathetic nervous system is activated. You're gearing up for all this metabolism. What else have I forgotten something? How about sweat glands? So when you're nervous, what happens? With those clammy hands, right? So what else happens when you exercise? You produce lots of heat. What do you want to do with that heat? You want to get rid of it. You can't allow the body temperature to increase so much. So, sympathetic nerves increase sweating. Anatomically, we have different origins. Physiologically, we have different effects. How is this manifested? At the bottom of the plate, you'll see cartoon diagrams which have kind of reiterated what we've just said. So on the left side, this is the preganglionic cell. Here's the ganglion and the postganglionic cell going for the sympathetic nervous system. For the parasympathetic nervous system, note that the preganglionic cell, very long synapses at the ganglion. The ganglion is within the tissue itself. This ganglion close to the thorax, this ganglion in the tissue. Sympathetic, parasympathetic. The neurotransmitter, both this ganglion and this ganglion, is acetylcholine. At the neuromuscular junction, it was acetylcholine that was the neurotransmitter. Muscle contraction was initiated by acetylcholine release from motor neurons. Those were motor neurons. We're now talking about the autonomic nervous system. This isn't going to skeletal muscle to produce contraction. These are going to skeletal muscle, to the blood vessels in skeletal muscle. They're going to lipid supplies, adipose tissue. They are going to the liver. They're impacting directly on the heart. So acetylcholine is the neurotransmitter at the ganglion in both cases. The big distinction between sympathetic and parasympathetic is the postganglionic neurotransmitter. That is the neurotransmitter here and here. For the parasympathetic nervous system, the postganglionic neurotransmitter is acetylcholine. The neurotransmitter for the sympathetic nervous system is norepinephrine. There's one important exception anatomically and neurotransmitter-wise to the sympathetic nervous system. And that has to do with innervation of the adrenal medulla. In humans, there is a small gland that sits above the kidney. So your kidney's down here, adrenal sits up above. Ad means above, renal means kidney. Anatomists called it an adrenal. In a dog, it's on all fours, it's not really sitting above the kidney, it's sitting anterior or closer to the nose. But it is a tissue which is comprised both of epithelial and nervous tissue. We'll talk in more detail about the adrenal gland. In lecture, you're going to see examples in the lab that distinguish between these two types of tissues within the adrenal gland. We're focusing today on the tissue that's inside, makes up the meat of the adrenal, called the adrenal medulla. Adrenal medulla cells are modified postganglionic sympathetic neurons. Now that's a mouthful. They're neurons, so they're from nervous tissue. They're modified because they don't have nice cell bodies, nice axonal projections, nice synapses. It's just kind of a cell body. So they're modified neurons, and they are postganglionic. So here is an example in which the preganglionic sympathetic neuron goes a long distance. This is the exception, right? Not the rule for the sympathetic nervous system. A cell body located in the spinal column, an axonal projection all the way out to a gland that sits above the kidney, innervates it. Acetylcholine is released here, just like it was at this ganglion, and just like it was at this ganglion. The difference here is that the postganglionic neurotransmitter is epinephrine. For all other sympathetic cells, it was norepinephrine. Now, there's like a hydroxyl group difference between them. It does produce some differential effects. But by and large, the action of epinephrine mimics that of norepinephrine. We call epinephrine a sympathomimetic. A sympathomimetic is a compound that mimics action of the sympathetic nervous system. Norepinephrine and epinephrine have many similar effects. Epinephrine is released into the blood. Norepinephrine is released primarily at the postganglionic, very narrow, very small window of action. This means that when you activate the sympathetic nervous system, you can get very fast, very immediate responses that can be shut off very quickly. But you have to remember that there's going to be some prolonged effect because this epinephrine gets released into the blood, circulates throughout the body. All right, so what else do we need to talk about? We have different neurotransmitters. We've got norepinephrine and epinephrine in the sympathetic nervous system. We have acetylcholine in the parasympathetic nervous system. Now, plate 30 talks about neurotransmitters and receptors. And before I move on to that plate, I want to go back and talk about cell receptors in general. How do cells receive or be receptive? So let's go to plate 12. What makes a cell responsive is its ability to perceive a chemical. We talked about the neuromuscular synapse. Motor nerve comes in, it releases acetylcholine. In order for the postsynaptic membrane to respond, in order for the muscle to be responsive to the acetylcholine, it needed a receptor specific for that neurotransmitter. Here's a generalized receptor. We've got membrane bilayer. Through the membrane, weaving in and out is this cartoon of a protein. So it's a transmembrane protein. It has on the extracellular surface a site that is specific for these compounds, neurotransmitters, chemicals, whatever you want. When this first messenger, or this first signal, binds to the receptor, what happens? Notice there's a shape change. There is a conformational change that occurs in this receptor. On the intracellular side of this receptor is some kind of effector, something that's going to affect or take the information that it's just gotten. That is, I've been turned on. I need to respond to this specific compound. And on plate 12, this effector is called the G protein. So the G protein is associated with the intracellular aspect of the cell. It's anatomically close to this transmembrane receptor. Cells are not just bags of soup floating around, things going mishmash. There is a very defined organization. We saw that certainly in the case of striated muscle, where you had this very tight organization. Well, even at this level, this G protein is located anatomically very, very close to this transmembrane receptor. So that when this transmembrane receptor gets activated by this chemical, it produces a change in the G protein. Now, G protein kind of sits on the inside, hanging on the cell, on the inside of the cell membrane, and it has bound to it GDP. I've talked about energy currency and ATP and ADP, adenosine triphosphate, adenosine diphosphate. Well, now this is, instead of an adenosine, it's a guanine molecule. And it's one of the ways cells can help produce specific responses. It's not ATP, it's not ADP, it's GDP. So right here it's showing the GTP molecule bound. When this gets activated, it releases the GDP and GTP binds it. And in that process, there is a movement. The G protein moves. So if I take, and they've drawn this little molecule like this. So here's my, they've drawn the G protein right here like this, right? And associated with that is the GDP. Well, when it is activated, it releases the GDP and binds GTP. So the G protein plus GDP move, and they move to activate another effector. In this case, the effector is another transmembrane protein. It is a protein which is an enzyme, and specifically it is called adenyl cyclase. When adenyl cyclase is bound by this subunit of G protein containing GTP, the energy from the GTP is used to activate the adenyl cyclase. Turn it on, if you will. So without any chemical messenger, without this, G protein is sitting quiescently on the inside of the membrane. Adenyl cyclase, inactive, not doing anything, but now you've precipitated a process. You've turned the system on. What does adenyl cyclase do? It's a cyclase. It's an ACE, so it's an enzyme. It's an enzyme which takes ATP and converts it to AMP, but in doing so it forms a cyclical molecule called cyclic AMP. Cyclic AMP has for decades been called the second messenger. This was the first messenger. It was taking a message to the cell. How did that get translated by the cell? It got translated by the production of a second messenger, cyclic AMP. Well, we see now that it's much more complex than that. There has to be a G protein. You've got to have GDP, GTP. You've got to have the adenyl cyclase. But once you produce cyclic AMP then becomes another effector in its own right. So here's cyclic AMP coming down and turning on protein kinases. So if you say adenyl, let's look here. Adenyl cyclase is turned on. It takes ATP and makes cyclic AMP. Cyclic AMP then goes to take an inactive protein kinase and convert it, so it's inactive, but it converts the protein kinase inactive to an active form. It's an enzyme. This enzyme can help turn on other enzymes. And one example would be glycogen phosphorylase. Sorry, that's right. So we have adenyl cyclase producing cyclic AMP producing protein kinase, and protein kinase then turning on glycogen phosphorylase. phosphorylase. Glycogen phosphorylase mobilizes glycogen. So why all this? Why is it so complex? Remember the definition of an enzyme. An enzyme is present in minute quantities, and it facilitates chemical reactions. So you do not need a lot of glycogen phosphorylase to take glycogen and make it to glucose. You don't need a lot of protein kinase to stimulate glycogen phosphorylase. You don't need a lot of cyclic adenyl cyclase. So each of these steps is an amplification. It goes back, in fact, to the G protein. There are more than one G protein per membrane receptor. So when this receptor is turned on, it turns on more than one of these G proteins. Say 5. Well, what you've done now is you've upped the scale by 5, at least 5, because for this one reaction, you produced 5 of these. These 5 go to 5 adenyl cyclases, an enzyme which stimulates production of cyclic AMP. It amplifies. It makes a lot of cyclic AMP. So you may have amplified the signal now by 100 times. Cyclic AMP turns on protein kinase, maybe another 10-fold increase in activity, amplification. And then the glycogen phosphorylase, another 10-fold. So each level of this enzymatic turn-on amplifies the signal. It is sometimes referred to as a cascade. And in biochemistry, they'll talk about the cascades, and they'll make you memorize all of the different enzymes that are involved in this process. Understand that it's a relatively fast process, and it is very effective at amplifying a signal. So your first messenger, you have receptor conformation. It's got to change shape. G protein gets activated. It goes to adenyl cyclase. Adenyl cyclase plays an important role in this. Second messenger, cyclic AMP, the protein kinase, and then the final target protein. And there are a number of different physiological effects. And you see down here there's enzyme activation. So that's the one we've been talking about, G protein's role in this. Gene regulation, muscle contraction, etc. Let's look at plate 13. Let me find plate 13. Talked about cyclic AMP as being an important second messenger. Now we're going to talk about calcium, the G protein. So, very similar. We have a membrane receptor. First messenger comes in. So here's the first messenger right here. Binds to the transmembrane receptor. A change occurs. G protein, again, GDP, activated. GTP binds. Now, instead of moving to adenyl cyclase, it moves to another enzyme, but not adenyl cyclase. It binds to phospholipase C. It is a membrane-bound enzyme which is responsible for the production of two important compounds. One is IP3, inositol triphosphate, IP3. Remember adenyl cyclic AMP, second messenger? Now what you have is you have IP3. IP3 really isn't considered the second messenger because IP3 has to do something else. So here is the kind of the schematic diagram. There's the phospholipase C. That enzyme then triggers the production of IP3. IP3 is responsible for the opening of calcium channels in the sarcoplasmic reticulum. This is a vesicle of SR. Remember skeletal muscle contraction? We had the sarcoplasmic reticulum. Inside the sarcoplasmic reticulum, we sequestered calcium. In a striated muscle, the nerve potential produced a change. It was a voltage gated channel. So the action potential responsible for opening that gate and releasing the calcium. Now we have an example in which a chemical messenger, so this is a chemical gated. When IP3 binds to this gate, the gate opens and calcium flows out. It is now the calcium that's the second messenger. In fact, it's more complicated than that. In order for calcium to be very effective physiologically, it has to be modulated by a protein. Whereas in skeletal muscle contraction, the calcium bound to what protein? Troponin? Well, in this system, the calcium binds to a protein called calmodulin. And that's that little molecule there. So calmodulin, a protein which modulates or helps the function of calcium. It is the calcium calmodulin complex that produces the physiological responses. Getting rid of calcium in this situation, there are two ways for calcium to go. One is to be taken back up in the sarcoplasmic reticulum. So here you see the calcium pump, and just like in skeletal muscle, calcium, there are pumps. These are active pumps that move calcium to the inside of the SR. There is also an external calcium pump. You can pump calcium out. That is also an active process. And the third way that calcium gets removed is by a sodium-calcium exchanger. So let's revisit ions. One of the key things that I'm going to ask you on exams are, which ions are in high concentration extracellularly, which ones high concentration intracellularly. So here's our cell. Sodium high on the outside, potassium high on the inside, chloride a little higher on the outside. Calcium high outside. If you pump calcium into the endoplasmic reticulum or the sarcoplasmic reticulum, it can be locally high, regionally very high there, but there's something like a 20,000-fold difference between the concentration of calcium here and here. If you're releasing this calcium, you can pump some back in here. you can pump some of it out here, but you can also take advantage of this gradient, this sodium gradient, to help pull calcium against its concentration gradient. This takes ATP, this takes ATP. The sodium-calcium exchanger, in fact, utilizes this gradient. So the energy that you get by letting sodium come in can be coupled to calcium being pumped out. Okay, so I want to talk about one more type of protein just so that you're familiar with it. And that would be plate 14. I've given you two examples in which cells have been linked to G proteins. Now I'm going to talk about other types of receptors that don't utilize Whoa. Give me a second. There we go. There we go. Tyrosine kinase receptors. Very large class of specific types of receptors, and they respond to a number of growth factors. And that's what I want is you to be familiar with some of the names of the growth factors. They're becoming very important in their role in the identification of disease processes. Epidermal growth factor. And you're going to find that these names have a number of similarities to it. There is platelet-derived growth factor. So again, it's a growth factor. Where did it come from? Platelets. Platelets are blood cells that you will learn about in lab. So these compounds are derived from platelets. Nerve growth factor. These proteins are very important in nerve growth. A lot of research of spinal injuries focuses on nerve growth factors. Fibroblastic growth factor. The last one, insulin. So here is a compound that is serving, goes to these tyrosine kinase receptors to activate it and turn things on. And we're going to talk about insulin and the role of insulin in blood regulation of blood sugar. The last protein that I wanted to know about is a transmembrane protein called the RAS protein. That is something that's very characteristic in proteomics today, is this RAS protein. And in this figure, the RAS protein here, I'm sorry I said it was transmembrane, it is on the inside of the inside of the cell. It is activated by these tyrosine-based compounds like nerve growth factor impinging on the receptor. The receptor then transmitting that information to the RAS protein. And what the RAS protein does, it uses GDP and GTP. So like the G protein, it utilizes those high-energy phosphates. When activated, RAS protein is important in response in producing the response of the mitogen activated protein kinase. Mitogen activated protein kinase. It's this compound that goes into the nucleus and increases protein production. So let's say you have a nerve cell. Nerve growth factor comes down, stimulates the receptor, which activates the RAS protein. The RAS protein stimulates another enzyme, a kinase, and it's called mitogen activated protein kinase. Mitogen, a production. Mitogen activated protein kinase moves into the nucleus to stimulate protein production, turn on the growth of this nerve cell. So it's helping produce proteins to increase nerve growth. There are a number of cancers associated with overactivity and lack of regulation of the RAS protein. 30% of human cancers are associated with the mutation of RAS proteins, of the genes that code for the RAS protein. The characteristic of cancers or tumors is unregulated cell growth. Cells keep proliferating. They keep growing. In some cases, this growth can be relatively benign. If you have a cancer of a lipid cell, all right, so the lipid cell keeps growing and growing. If it happens to be around your gut, you may get this very large growth, very easy to remove. If it's a RAS protein that's involved in nerve cells, you might now have a neuroma. Nerve cells going unregulated. The problem is they start occupying space and there's no control over them. So it turns out that this protein plays a real significant role in cancer and one of our big issues. So there are cell physiology courses that go into this much more detail, but what I would like to do is focus on other types of receptors in the autonomic nervous system. But we'll take a break now. Whoop. Okay, are we good? Do you fall asleep?